impressive, but clearly not impressive enough to make the PowerPoint work. But anyway, we've got it now. We've heard a lot about Section 3D. We've heard a lot about evergreening, whether or not evergreening is justified. You have your patent application for a new chemical entity. Should you then be allowed to come along and claim all these other things afterwards? I'm just going to assume that you've decided that you want to do that. As far as I can tell, every pharmaceutical firm which I've ever had contact with has decided to do this themselves. The innovators do it, the generic companies do it. Everyone wants to join the party. So I thought it might be helpful if we just focus on the nuts and bolts of how you do that successfully. If you want to file patent applications on things like for enantiomers, polymorphs, downstream medical uses, can I give you some hints and tips on the best way of achieving that? Because it's not always straightforward to get them granted. Okay, I'm going to launch straight in, talking about enantiomers. And the first message is that claims to specific enantiomers can be upheld in the EU at least and in the USA. I assume everyone knows what I mean by an enantiomer. If you have a drug which is sold as a mixture of enantiomers, a racemic form, and you later come along with a patent application, later a patent for the specific enantiomer which has got particular therapeutic efficacy, claims along those lines can be upheld. And we've got two examples here. Generics v. Lundbeck with the escitalopram drug, very famous, and that patent really has been upheld throughout the world. Generics v. Daiichi, that's the oflafloxacin, levofloxacin case, again, the enantiomer was upheld. And we've got a quote here from Lord Justice Jacob. He's a very well-known judge in the UK, very distinguished. Only a curmudgeon will say that there is no invention here, he says, talking about the enantiomer. I looked up curmudgeon actually before I did this talk. I find that it means a bad-tempered or surly person. So clearly he would assist the temperature of the debate here in India about Section 3D. But that's what he says about it. It's clearly patentable, and you're a bit strange if you don't think there's a patent. Now, one thing to say is that necessarily when you're looking at claims which are coming to be litigated now, they have a priority date that goes back 20 years. So it's necessarily the case that you're looking back in time and you're saying it was inventive at that point. The question are enantiomer claims still patentable now is a harder call. I mean, everyone knows that if you have a racemic mixture, there will be a eutamer and there will be a distomer. And by that I mean one of the enantiomers will be more active than the other. That's very well established and very well known. And so one could say that these days it's going to be much harder to demonstrate that a claim to a specific enantiomer does enjoy inventive step. My answer to that will be that, as far as I can tell, in my experience, patent offices are still granting these claims. If you come along with a claim to a specific enantiomer and you show that there is some advantage associated with it, be that improved efficacy, be that reduced toxicity, even though you may say, well, perhaps in principle claims shouldn't be allowed for this sort of invention, they do seem still to go through the European Patent Office and the US Patent Office. And how do you draft your application to facilitate those cases going through? Well, in both in the escitalopram case and the levofloxacin case, there was some difficulty at the priority date about achieving the resolution. Now, you may say, well, these days chromatographic separation has come on very well. Chiral columns are very well known. Actually, in practice, there would be no difficulty in achieving the resolution of any given enantiomeric pairing. Well, that may be the case. But when you're drafting the description, it will help if you can tell a good story about the process of separating the enantiomers. If you had to do some trial and error to get them to separate effectively on a commercial scale, put that in the description. If a particular separation technique was found to be successful, perhaps despite an initial belief that it might not be, again, put that in the description. It will help the case go through without too much argument. Obviously, you are going to need some advantage associated specifically with the enantiomer you're claiming. I've said unexpected properties. Well, ideally, you want something that's unexpected. So in the escitalopram case, the distomer, the R-citalopram, was not only inactive, it was actually inhibitory. So when you made the resolution, there was a massive increase in activity, which was unexpected. If there's anything as unexpected as that, then that will be very helpful to put in the application. If you haven't got anything as good as that, you just have some sort of therapeutic advantage, this one is more active than the other, that might be enough with a following wind. So put that in and write it up. and then identify particular impurities and their maximum levels. Now, how do you actually go about writing the claim to an enantiomer? Well, obviously you claim a compound which is, and then you write down the enantiomeric form. That may or may not be good enough. It should be good enough in most cases. But I think it would also help if when drafting your application you put in language to help you define more carefully exactly what it is you're claiming. So a compound of this formula, well, okay, if we're going to be really picky, I could say, if I'm just claiming, say, escitalopram, a claim to a particular chemical compound, according to the normal rules of claim construction, covers that product under all circumstances and in any environment. So why doesn't it read on to the enantiomer present in the previous prior art racemic mixture? Well, one could take an interpretation like that. I don't think it's a particularly valid or worthwhile interpretation, but I have experienced objections from patent officers along those lines, particularly in the less sophisticated patent offices. They actually want to see language in the claim that says, look, it's substantially separated from the other enantiomer. I would have thought that's obvious and implicit, but I think it's still worthwhile having that language in the description somewhere, or somewhere in your application, so that you can put it into the claim if necessary. You can also consider this consisting essentially of language, a compound or a composition which consists essentially of the particular enantiomer, and then if you found a way of producing the enantiomer, then obviously claim that as well. It may also help in some jurisdictions if you include actual numerical figures for enantiomeric excess. So the enantiomeric excess of the compound you're claiming is at least 95%, preferably at least 98%, more preferably at least 99%. And that may help you to write in an appropriate limitation if that becomes necessary. It's very easy things to do. You can easily write a template specification with all of this language in it, and then you just roll it out each time. But I have seen so many applications being filed without this language, which doesn't really give you anywhere to go if you get into trouble during prosecution. Just moving to the USA, this is the escitalopram case in the USA, Forrest v. Ivax. Effectively, they came to the same conclusion. They upheld a claim to substantially pure escitalopram, and their thinking here was much the same as in Europe. The primary reference, the prior art, discloses the racemate. It doesn't describe how to achieve the separation. There was some difficulty, some invention in actually achieving the separation, and there was also, I think in the next slide, we say there were some unexpected results associated with the specific compound claimed. So we have a fairly consistent view from the USA and from the EU on this topic thus far. A slightly less happy story is the Aventis v. Lupin. Again, this was came via paragraph 4 certification. I don't want to get into the paragraph 4 procedure, so I just want to look at the merits of the case. But there they were claiming the 5S stereoisomers of ramipril and derivatives. And the prior art there was a mixture of the 5S and 4SR enantiomers. There were separation methods taught in the prior art. But Aventis came along. We said, look, we're claiming a particular diastereoisomer, the 5S compound, and that is unexpectedly effective. It is much better than the next most best isomer. And that was an example of a claim that was actually shot down. One of the reasons, and I think the main reason in my view from reading this case, is that the court said that you haven't compared with the closest prior art. Your comparison needs to be with the compound disclosed in the prior art. In the prior art, it discloses a mixture of the 5S and 4SR isomers, and you haven't made your comparison against that mixture, and therefore we're not going to take the activity data into consideration. So I'm going to move on from enantiomers now to polymorphs. Polymorphs are obviously well known to you all. A polymorph, of course, is a different crystal form. Just to keep you awake, I'll give you an interesting story about polymorphs, how they may have affected the history of Europe indeed. I believe it was Napoleon's army marching into Moscow, and they had some very shiny tin buttons on their uniform. They polished up, looked very nice. Of course, as it got colder and colder and colder, there was an allotrope. I suppose it's tin, so it's not a polymorph, it's an allotrope. But they changed to the different form, and they crumbled to dust. And rumour has it, I don't know whether this is a myth or rumour has it, that the troops felt that that was a sign of divine disfavour. Morale plummeted, and it's given by the historians as one reason perhaps why the invasion of Moscow was unsuccessful. So there you go, a little anecdote about why polymorphs are interesting. But in the field of drug discovery, of course, a polymorph is an opportunity for evergreening, if we're going to put it like that. How do we go about that? Well, let's start with the European Patent Office. One of the leading cases, 77708, this has been discussed before. Effectively, this case is saying that there is no invention simply in identifying a crystalline form. So if it's known in amorphous form and you found that you can crystallize it, that's not an invention in and of itself. If it's known in a crystalline form and you've made it in amorphous form, that is not an invention in and of itself. You need some sort of unexpected therapeutic or technical advantage associated with a new form you're claiming. And you can't just say, well, it's crystalline so it dissolves better or something like that. There are known advantages to forming crystals as compared with amorphous forms. There are known advantages of amorphous forms over crystal forms. Simply switching from one to another and reciting the known advantages is not going to be good enough. Nevertheless, it is still possible, in my experience, to get polymorphs granted by the European Patent Office. Obviously, there's going to be some doubt about whether these cases will be upheld, but we have had some success in upholding these claims in European courts, Leo v. Sandos being the recent example, where the High Court and the Court of Appeal of the United Kingdom upheld a claim to the calcitripol monohydrate in a particular polymorphic form. So it's worthwhile getting them. How do we go about drafting a claim for a polymorph? Well, first of all, let me just go up one. As I said, we do need some sort of therapeutic or technical advantage associated with a particular polymorphic form. Now, whether that be some sort of improved solubility as compared with the other crystalline forms known on the market, some sort of improved packing ability, improved flowability, something that gives you a technical advantage. You need to refer to that and explain that in your specification. If you don't have that, then you're not going to get a patent granted. So that's an essential first step. I wanted to start off by looking at this case because having got to that essential first step, as I said, this presentation is supposed to be about the nuts and bolts of how you put these specifications together. So this is a case in the USA where a polymorph was cut down. They said you couldn't have a patent. There were two reasons for that. I mean, the first point is a blindingly obvious point. It wasn't properly distinguished for novelty purposes. Well, I guess that will be obvious to every competent patent professional. If you've got to claim a polymorph, you need to show that it's different from the polymorphic forms which were disclosed in the art. But look at Claim 8. This is a little bit of a more interesting reason. They claimed a pharmaceutical composition comprising this polymorphic form, whereas at least 80% by weight of it is in crystalline form 3. And the examiner said, well, that's all very well, but it's well known that polymorphs may undergo transformation during the formulation proceedings. When you put it into a tablet, it might change form. And that's particularly the case because, usually, or not usually, often, if you're trying to claim a new crystalline form, and there are crystalline forms in the art, often you'll be looking at a sort of an improved dissolution, improved bioavailability, to justify your claim, to justify the invention. Well, if it's got improved dissolution, improved bioavailability, that's generally because it's a higher energy form. And so what the US examiner is sort of saying here is, well, if it's a higher energy form and you grind it up and put it through an extruder, it's probably going to revert back to the lower energy crystalline form, and I don't believe that you can make this, so there's an enablement problem. Now, hold that thought, because those objections do come up in the USA. I experience them quite often. But it does seem that they can fairly easily be sidestepped in practice. Okay, so what do you do when you're drafting your application? As with the enantiomers, tell a good story about the process of producing the polymorph, again, particularly if there's trial and error. Oh, we had to mess around with various solvents. It might be utterly routine work, but try and write it up and make it sound as difficult as possible. I suppose that's just general tips for drafting patent applications. Once you've said how difficult it was, you need to define the crystalline form, and you need to distinguish it from that which we've done before. So you can do that in various ways, of course. You can define it by its X-ray diffraction pattern, its Fourier transfer IR spectrum, or its Raman, the unit cell. There are various different ways of doing that, and put as many as you can in. So, for example, if the scientists come to you with XRPD data and say, look, we've got a new polymorph, tell them to go away and do the IR spectrum as well. It doesn't take very long. It's not very difficult. Put as much in your application as you possibly can. When you've put it in your application, and again, forgive me if I'm saying the obvious, but I do see so many applications which don't have this. Don't put all the peaks into claim 1. Don't say compound of crystalline form of compound X characterized by the following XRPD spectrum, and then put all the peaks in. It will be a complete nightmare to demonstrate infringement if you do that. So you can just look at the top 3 peaks, an XRPD spectrum where the top 3 peaks are A, B, C. Preferably, the top 5 peaks are A, B, C. More preferably, the top 10 are, etc., etc., etc. So you can build up in that way. Just look at the fingerprint peaks, rather than putting everything into claim 1. Again, very obvious point. And then you can also, I said, try and get as many different data types as possible. Again, a very obvious point, but don't put them all in your claim. Don't say it's got to have an XRPD spectrum like this and an FTIR spectrum. You can use the and/or language or the or language. So a crystalline form of compound Z characterized by the XRPD pattern with one or more peaks, etc., or a Raman spectra, or an FTIR spectra. So you can put them in as alternatives and then build them up if necessary afterwards. Again, this is the same point as the enantiomer point. You might want basis to say that the crystalline form is in pure form, substantially pure form of the drug. And then you might have comparative physical data say, well, actually, we ran the synthetic process in the prior art and we get this different spectrum on the prior art product. So there's no issue with novelty. And I would recommend that if you have that data, you put it into the application as filed. It'll make things much more smooth during prosecution. And then to demonstrate inventive step, as I said, you need some sort of technical advantage, and obviously that's got to be in your application as well. I think that goes without saying. Again, this may be obvious, but do specify all the information you need to repeat the characterization. I've had a handful of cases recently that I have been asked to prosecute and defend, which very nicely have an XRPD spectrum, and very nicely say, well, this is very different from the prior art. But the problem is they don't specify the wavelength at which they did the X-ray diffraction. And the European examiner, I think quite rightly, is saying that's fundamentally unclear. It's both unclear under Article 84 and insufficient, because obviously the 2 theta values will change depending on the wavelength at which you measure them. That might well be a fatal objection. I have some ideas about how we can address it. But if you haven't put the information in the specification as filed, it can be fatal to the case. In some cases, examiners have asked for the anode material to be specified. If you've got a DSC curve, a decomposition curve, you need to specify the heating rate, the reference material, and the sample size. Anything which could change the number needs to be specified in the application as filed. Again, forgive me if I'm coming out with points that are obvious, but as I say, perhaps it's sometimes useful to do that, collate them all together so we can hear them. And then coming back to this enablement issue, if you are basically claiming a higher energy crystalline form, how do you deal with the enablement objection? I mean, this is just my experience. I have found that if you put this paragraph into the description, the US examiner is much less likely to raise the objection. And then if the objection is raised, you point to this paragraph in the description, the objection often goes away. Just say the claimed crystalline forms a high melting point solid with residual solvents within permissible limits, very well suited for formulation. Now, of course, if you have experimental evidence of, well, we've done the formulation and we retain the same form, all well and good. But if not, then you can put this sort of boilerplate language in, and it does make life a bit easier. I'm going to spend just 6 minutes or so speaking about medical use claims. I think we've heard a lot about them already, only because I noticed that was in the title of my talk, and I haven't got any slides on it, but never mind. We talked about new uses of a known drug, novel dosages, new patient groups, new modes of administrations, and sometimes even where you have a new etiology for treating the disease. I just want to pick out one of them because I think it could be quite interesting, and that's the new patient group. And the case we were referred to earlier was the PIGS decision from the European Patent Office back from the 1980s. method of treating Ajetsky's disease, I think it was, or vaccinating against Ajetsky's disease in seropositive pigs. Now, the point there is that the prior art talked about seropositive pigs and the invention was about treating seronegative pigs, or the other way around, I forget which. But there was no overlap between the patient groups. And then haemophilic mammals, non-haemophilic mammals, subsequent case law, again, no overlap between the patient groups. What I think is interesting is we are now seeing a line of case law from the European Patent Office where claims are being upheld which specify a patient group which is a subset of the patients treated in the prior art. And an example of this is the HCV case. I think it's T309904. It was for the use of a particular blend of compounds for treating HCV, hepatitis C virus. And the prior art said, use these drugs to treat HCV, full stop. That's what the disclosure of the prior art was. The applicant came along and says, well, okay, well, I found that if you use the drugs to treat HCV in patients which are, firstly, antiviral naive, they've not been treated before, secondly, they're suffering from a particular form of HCV, and thirdly, they have a particular viral load, certain million copies of the virus per milliliter of serum, it's particularly effective. And because it was particularly effective against those specific patients, the Board of Appeal upheld the case. They accepted that this was a subset of the patients previously treated. They also accepted that it was a big subset, perhaps as high as 50%, yet they nevertheless upheld the claim. And the reason why this has attracted so much attention, excitement, is that it's becoming quite commonplace now to collect data about patients during the phase 3 clinical trials, particularly in the oncology field. You may be collecting, for example, genetic information about the patients that you're treating in the phase 3 clinical trial, then you find the drug is effective. And companies are then beginning to mine the phase 3 data, saying, okay, well, let's see whether we can find, is it particularly effective against red-headed patients? Or, I mean, obviously that's frivolous. It may not be effective against markers like that. But if it is, then you are able to repatent that effectively and almost reset the patent term starting again, because if you can, of course, say that, okay, it's particularly effective against red-headed people, I'm going to claim that, I'm going to put that in my SMPC, Summary of Manufacturing Product Characteristics. A generics company who wants to launch a generic product wouldn't necessarily perhaps have to copy the SMPC in many jurisdictions, and they have a prima facie infringement problem. Now, I don't know how that's going to pan out. It's just one line of case law at an early stage, but that's of some excitement in the field at this time. Okay, I'm going to leave it there. I think the plan is that we'll take questions on the medical use aspects and the pharmaceutical aspects, and then come on to the last topic afterwards.
Microphone. Hans Brandy Dorn from Berlin in Germany. I have a question to Santosh regarding this specific Canadian jurisprudence regarding selection of species. Is that applicable exclusively to this specific topic, selection of species, or is it a general jurisprudence regarding any type of selection of, say, a certain range, dosage, size, weight? If that would be the case, that means that I would have to demonstrate in my patent application that for the entire range I'm specifically mentioning in my application, I would have to give supporting data.
Yeah, unfortunately, the short answer is yes. The way the courts are looking at these selection patents is very much like that they're looking for support for that entire range. If I'm not mistaken, one of the cases that I have in that table dealt with a range of a compound that's able to be put into a formulation, and the claimed range was, I think, from 20 to 70%. And during the trial, it came out that the maximum that they were able to achieve was 50. So it was clearly, they overextended the claim. Obviously, for that reason, there was no data to support the entire range as well. So the short answer is yes. If you want to stake out a claim, then you have to have some data to support the range. Now, again, going back to this issue of demonstrated versus predicted, if you demonstrate utility over a certain portion of the range you want to claim, you're still entitled to that broader range as long as you can have that reasonable prediction that it would apply. And this is where the disclosure requirements come in. In order to substantiate that predicted utility, you have to, again, as Ravi mentioned as well, tell the story. So explain what your basis is to make that prediction, and explain what the line of reasoning that you have to extend it. Now, again, the example I gave of that case, that was unfortunate because they had overclaimed, and for that reason the claim was deemed invalid. But as long as you can substantiate as a prediction, then you're fine.
Hi, I'm Durgesh from K&S Partners. My question is for Ravi. You touched upon special patient groups. Now, how it is important to show that a particular compound or a composition worked in special kind of patients, patient population. Can you touch upon how important it would be to show negative data in this regard to probably show that it works more efficiently in certain population but not as efficiently in some of the other populations related to that?
Yeah, you'll definitely need some comparative data to justify a claim along those lines. So, for example, if the claim says, well, this is useful as an analgesic to treat headaches, and you're saying, well, actually, I want to claim it particularly in Asian populations over 30 or between 30 and 35, you need to show that it is better in those patients than it is across the rest of the scope. Otherwise, you just carved out an arbitrary group. So that's why I'm saying that this sort of invention, if you put it out in quote marks, is often done at the clinical trial stage, because that's when you tend to find at the phase 3 clinical trial, phase 4 clinical trial, you tend to pick out differences of that nature.